We're going to do a little bit of a deep dive, in as much as we have a few minutes to do that, in the sort of molecular space, targeted therapeutics, gene editing, things like that, and kind of walk you through a little bit of that. We are missing, unfortunately, one of our panelists, John Madison, who's an old friend of mine, and he was going to give us a little bit. He's a physician, works a lot with data at Kaiser, and he was going to give us a little bit of a physician's or a clinician's point of view. But I think we're going to have a lot of fun here over the next few minutes and learn a lot about what's happening in this really exploding space. As a journalist, I've been covering genetics. I first got sequenced in 2001. Actually was one of the early people to do that. And it's taken a while. These things always do. But we're at a period right now that's quite exciting. In fact, I want to reframe the panel topic just a little bit, and because I like to talk about health. I mean, medicine is what happens, or at least when you have medical treatments, when you're not as healthy as you would like to be. But this space is really as much about keeping you healthy now. I call it radical wellness, using massive amounts of data. And it also only starts with genetics. In fact, in a talk I give, I have a slide that says, genetics is so yesterday. And it's not that it's not important, it's just that it got so much attention for so long, when in fact, it's all about what you call multi-omics. And you've heard a little bit about that in the previous talks. The connectome is actually even part of that. But there's the metabolome, the microbiome, even the epigenome, all these different networks and pathways and types of molecular health pathways. So we're going to talk a little bit about all that, but I'd like our panelists to each take a couple minutes to tell us what you all are up to. And we start with Clayton Lewis.
Great. So what Arivale focuses on is helping individuals, one, optimize their wellness, radical wellness, and two, avoid transitions to diseases. And the way we do that is first we start by creating a dense dynamic data cloud for each individual. We look at genomics, understand what is your predisposition. We look at over 80 clinical markers because that's actually a reflection of your lifestyle to date. We look at gut microbiome. We measure your saliva 4 times during the day to get insights into your cortisol levels, and then of course collect a lot of data. Now, what we know is data actually paralyzes people. So what we do is we organize that into health dimensions. Each of you in the room today would have your health dimensions stacked ranked on where there's the most important opportunity for you. Heart health, diabetes risk, aging, inflammation, nutrition. And then you'd be assigned a coach, a registered dietitian, backed up by a clinical team, backed up by a physician who'd give you actionable recommendations each month to optimize your wellness. Then in 6 months, we'd check your blood again and see if we've made progress.
That was great. In fact, I didn't really designate where you guys sat, but this is kind of a nice order because in a way, Clayton is talking about the end product, at least what we see. And I've actually been tested by Arivale. And in fact, I don't know if you all are familiar with a book I wrote a few years ago called Experimental Man, where I took every test on the planet. And I've talked about it at previous FiRes. But I'm actually being tested again. And one of the curious things about this is I collected about 15 terabytes of data between 2001 and maybe 2008. And then the book came out in 2009. In the last year, I've collected well over 50 terabytes. I kind of lost track at this point. So the ability to collect this data is extraordinary. I mean, those technologies have moved along very quickly, and you're getting companies like Arivale. And I love some aspects of what they do, especially the coaching he mentioned. You have somebody that actually calls and walks you through this data. We can talk about more of that in a minute. But so Ben is sitting on that side of Clayton. He's the guy that helps us understand what's going on with this data. And is, again, somebody that's been almost Consumer Reports or test driver of all this data for years, it hasn't made a lot of sense, I have to admit. And even now, I mean, probably 99% of that vast amount of data you collect on one guy. The real bottleneck is what the heck does it mean? And I used to give a talk called Biobabble, Too Much Data, Too Little Understanding. But even that is coming along. And I just want to, Ben, tell us what you do at Berkeley and how you are helping us with this dilemma.
Sure. So I'm the department head for molecular ecosystems biology at Berkeley. And what that means is we try and develop an understanding from molecules up of living systems. Some of those are very complex multi-organismal systems, like a forest. Others are very complex multi-organismal systems like a human, along with all of our gut microbiota and lung microbiota and everything else. For these systems, what's really been transformative in the last 2 decades has been our capacity to make measurements at many, many different scales and many different types of molecular measurements. For those of you who remember the central dogma of molecular biology, DNA codes for RNA codes for protein, since the late 90s, we've been able to use microarrays and then, of course, later sequencing to see the RNA output of the genome. And so genes are expressed and exert their effect when they are expressed, when the DNA is transcribed into RNA, and then that RNA is subsequently transcribed into protein. Protein can be an enzyme, can be a structural component of the critter, whatever it is. And you have the same genome in every cell in your body, right? The DNA is the same in your eye as it is in your nose, as it is in your bones. And so why do we have such wondrous diversity of tissue? Well, that's all to do with gene regulation, right? Which genes are on or off, in which pattern where. And of course, it's not just on and off, it's all rheostatic, it's all quantitative. And so in just the last 5-6 years, really started to be able to get single cell pictures of how complex consortia work, whether that's a microbial consortia in a biofilm in a freshwater lake, or whether that's mammalian cells organized into a tissue, part of an organ system. We've really started to be able to explore those at a molecular level. And of course, it's not just RNA and protein. And protein has made leaps and bounds. Proteomics has gotten much better just in the last few years, as far as the number of proteins we can measure and quantify in a system. Now, metabolomics has come an enormously long way, right? If we take a human blood, urine, anything, you name it, take a human sample, and you run it through liquid chromatography, fire it into a mass spectrometer, and then ask what chemicals are actually present. The answer is going to be upwards of 10,000 that we can detect, right? And for virtually all of those peaks in a mass spec signal, we have no idea what they are. If you know what 500 of them are, if you know what 500 of the chemicals are, that's great, right? And so there's a lot of projects now underway to characterize more of the microbiome. So the microbiome is, the metabolome, the chemistry. The metabolome is today where genetics was and where genomics was in the early 90s. But it's coming fast. And so what we do is try and weave together many different types of omics measurements, even imaging stuff, whether that's medical imaging, MRIs, or whether that's hyperspectral remote sensing. Try and layer all of these together using statistical machine learning for data integration, and then try and use various perturbation techniques to perturb particular elements of the system, try and figure out how it works. And one of those tools is targeted gene editing.
Yeah. One thing that's really fascinating about all this is, too, the genome is somewhat static. We know now it's not entirely static, but part of what you're talking about is that they're dynamic systems. They're changing constantly. And, both of you, that must be an extraordinary challenge to keep up with this. I mean, you're testing people every 6 months, but you could be testing people, continuously, because it's changing all the time when you get into the proteome and the metabolome.
Well, and what's fascinating, of course, is looking at the correlations between all of these omics. Because in addition to the data I shared with you on a discovery basis, we also collect proteomics and metabolites. And because we've had some people in our program now upwards of 3 years, and we have blood draws every 6 months, very unfortunately, we've had a number of people who've obviously transitioned from wellness into disease states. I mean, right now in this room, a number of you have diseases racing through your body. And some of those will present in months, some in years. And of course, that's what our medical system is designed to do today, is to treat symptoms as opposed to keep you well. Well, very unfortunately, we had a member who came and shared that she had stage 4 pancreatic cancer. And of course, that's usually fatal because of when it's diagnosed. And so we went back on a non-hypothesis basis, looked at all of the data that we had for this individual, because we had 6 blood draws. And not surprisingly, at the very first blood draw, she had a protein that was materially out of range compared to everyone else within the program. And as the scientists dove in, that protein was associated with the function of the pancreas. So the big idea, and obviously it's an N of 1, and I'm not proposing that that means something other than the signals, But the big idea is that imagine if you understood your genetic predisposition. Imagine if you understood the omic that you needed to be tracking that was an indication the disease was triggered, and actually could target that based on looking at this non-correlation hypothesis of what are the relationships between microbiome and metabolites and your genetic predisposition.
No, I mean, that's this term radical wellness that we came up with is really, it's radical in the sense that we don't do this very well. But you do want to find that place where you go from a state of healthiness to a state of disease. And there is a tension all the time in your body, and your body can deal with much of this for most of your life. But if you could get farther and farther towards that moment, and the exciting thing is that the data, well, to help us out here a bit. I mean, are we getting to the point where we can understand that better now?
It's getting a lot better. The integrative analysis and being able to have, as you said, multiple time series, multiple omics on the same samples is what's been really transformative. So when we have, if you have just gene transcription, you see what genes are doing. That's nice. You can connect it to phenotype. You can say, okay, these genes are connected to phenotype. You can try and interpret that by saying, oh, we know these genes are involved in these biochemical pathways. But if you have the metabolome as well on the same sample, you can model the metabolome as a function of the transcriptome, as a function of what genes are doing. And then you get very direct insight into biochemically what's going on. You can then take that model that models, basically chemistry, the output of cells, as a function of what the cells are doing with transcribing their genomes. And you can take that model and you can regress phenotype. And that gives you a lot more definitive insight into what's going on with a system, medically, environmentally, whatever. And so the more omics layers you can put together, the more profound the insights will be. The remarkable thing about omics technologies in general is that you are measuring everything. That's what omics means, right? You're measuring a universe of things. And virtually all of the time, you don't care about 99.99% of that universe. And so the first task in omics is feature selection. It's finding the few things that matter or drive the process you care about. And having multi-omics modalities on the same samples has been absolutely transformative for our ability to zoom in on the stuff that matters.
Yeah, and let's jump in a little bit to the more medical side just for a second on how this applies. Gene editing, that's sort of a buzzword right now. I just did a story for Wired in April on the first human synthetic genome that's being done at Harvard. And it was crazy in good Wired style. They actually used my genome. But apropos to the conversations earlier about redesigning humans for space and to deal with radiation, things like that, this is actually happening in this lab. And it's not just my genome, it's not a copy of it. It's actually improving, and believe me, there are a lot of improvements that you could make. But this is going directly from a microbiome experiment and paper that was published, and this is George Church's lab at Harvard, where they were able to exchange a few nucleotide, which were the code letters in DNA, in different codons in a different pattern in bacteria, and make the cell that you then put this genome in impervious to viruses. And that has some interesting issues because that may or may not be a good thing all the time, but since a lot of our genome actually is vestigial viruses from the past, which might be important. But they immediately just jumped right to the big kahuna and went to humans. And they're working on that right now, and the first chromosome they're working on is the Y chromosome. Not because, actually it's a woman that's running the project in the lab, but the Y chromosome is so tiny and puny and simple. So I'll just leave it at that about the Y chromosome, but that's easier to construct. But Ben, tell us a little bit about that universe where we are. Where does that apply to potentially human therapeutics or gene editing? Maybe you can even explain what that is.
So gene editing for human therapeutics is a thing right now. There's a number of clinical trials in quite a few countries. It's been going on for a lot longer than most folks appreciate. The first big gene editing technology was zinc finger endonucleases, as far as being able to go into a living organism and edit its genome in situ. If you can get instructions into a cell and you can damage the native DNA of that cell, there's a good chance that you can leverage the cell's own what's called homologous recombination, which is so if you get a double-stranded break in your DNA, chromosome breaks apart, terrible type of DNA damage, very, very worrisome. What your cell looks for when that happens, it's got a very complicated, very elegant, and reasonably well understood system to try and identify another piece of DNA that looks like either half of the thing what just broke, and then stitch it back together and replace any missing bits from the template, right? And then we have 2 copies every chromosome, so usually that template's the other chromosome. A lot of double-stranded breaks are repaired perfectly, some not so much. And so the first big thing, a little company, a little startup in California called Sangamo Biosciences then, Sangamo Therapeutics now, they figured out how to sort of tame zinc finger endonucleases and make them target a very specific region of the genome. They never got down to single-site specificity. It would actually chew up the genome a little bit more than you might like. But they were able to introduce foreign DNA and replace a gene in a human. And they've got a clinical trial right now for Hunter's disease. And they've just fixed up 2 kids' livers, right? They create a virus that infects the hepatic tissue pretty efficiently. And the virus injects both the template DNA of the healthy copy of the gene and the zinc finger endonuclease. The cell's own machinery within the liver transcribes the endonuclease, translates the endonuclease into protein, gets imported back into the nucleus, chops the DNA, cell uses the template. It's not successful in every cell in the liver by any means, but you go from not being able to process a certain type of sugar to being able to process it, from having a very abnormal lifestyle to having a normal lifestyle. And so those technologies are real right now, but there's a big leap between that and editing the germline, right? Those kids, that's not heritable. If they have kids, they're still going to pass on the mutant messed up gene. And so the big question now is, of course, editing the germline of humans, changing us when we're a small.
That's what you pass down to your progeny. Yeah, there's your cells, and then there's cells that you might pass on to your children. Yeah.
So folks are experimenting now with a lot of monolocus diseases where there's just one gene. And it demonstrated you can cure mice, all sorts of stuff. If you're a mouse, this is a great time to be alive.
How many times have we cured cancer in mice?
A lot of times.
Yeah, right.
a lot. Yeah. No, my mice are doing great. But that technology, the basic proof of principle, is being spun up. There have been examples in dogs, in pigs. I think there's a new one in sheep coming out soon. So that technology is scaling, and we will for like single locus, simple congenital diseases. Those are going to be very fixable.
No, it's pretty fascinating, and we could get into so much of this that we don't really have time to. But the understanding of genetics. I also wrote a book on the science of longevity, and the one area we've already increased lifespan by quite a bit just by eating well and good hygiene, things like that. But there are people working on genetics, and I was a huge skeptic of this at first, but doing things like what Clayton's doing, I mean, that's the baseline, but we might be moving into being able to tweak these genes, which we've done in some animals, to potentially slow aging or some other aspects of that. But I wanted to give you all time for some questions, so maybe if you want to start heading towards a microphone. And I want to move back again to what this means for us as humans and individuals. And having done Arivale, everybody always asks me, well, did anything change, or did you change anything? And fortunately, I'm pretty healthy, but there are a couple. There's a metabolite called TMAO, and this is actually produced by microbiome in your gut. And I guess the underlying mechanisms aren't well understood, but if you have a high level of that in your blood, it suggests a higher risk for heart disease. And you can deal with this. It's also correlated with eating red meat. Correct me if I'm wrong about this. And so my coach, whose name is Ginger, by the way, and she's wonderful, she's been coaching me through, and I'm on now my 3rd blood test, but my TMA score went from pretty dangerously high, I mean, at least kind of alarmingly high, it was about 97 percentile down to normal. And I just did that. I thought I was eating less meat, but I almost cut out red meat. And so something's happening with my microbiome. And so that's a case, just a tiny little incident with one person on how a lot of these different omics kind of appeared all at once. But
I could share my story quickly. So when I launched the company, so my co-founder is Dr. Lee Hood. And when we launched the company, we did an IRB-approved study of 108 individuals. And I joked with Lee, I'm going to be the healthiest person in the study. I run Ironmans. I'm president of the board of Harvard Medical Center. I spent my whole life trying to be healthy. So my data comes back, and the first thing my coach says is, Clayton, you're pre-diabetic. And I'm like, okay, there's been a data switch. That could not be. Well, I had gone on a paleo diet about 6 months before, thinking the paleo triathletes seemed to be faster. And what she helped me understand is my genetic makeup, I have variants where I actually cannot process a paleo diet. I need rich, dense, complex carbohydrates in every single meal to normalize my blood sugar level. So think how counterintuitive all these people doing these fad diets, not understanding their genetic predisposition. Second thing she told me is I had toxic mercury level, so high that it was going to do neurological damage over time. Now, how could this be? I get a physical every year, I'm highly engaged. Well, of course, your typical physical doesn't look at toxic or heavy metals. So once again, I have variants, a lot of you have them, where I don't process toxins as well as some people. But the value of a coach, she said, well, do you eat a lot of tuna sushi? Well, I'm a salmon guy. Well, how old are you? And at the time I was 57, and she said, well, do you have a number of old fillings? And for a decade, my dentist had said, you know, someday we should replace these fillings. And I'm like, someday we should. Well, boom, went in and got those fillings replaced, and 6 months later, my mercury level normalized. But that's sort of the power, is understanding genetic predisposition, but genes are not your destiny, understanding what you're going to move, your clinical analytes, and the recommendations on one level are simple: eat less red meat. A high TMAO score has both short and long-term cardiovascular damage, but most people don't have insights into what's going on in their microbiome and have no clue to how to actually make it actionable.
Great, you're telling me the short-term damage?
Yes.
Oh, great. Nobody mentioned that. Okay, so if I drop dead here, I guess that's...
Exactly.
Anyway, let's grab some questions. We have some people out there, right? Why don't I start over here?
I'm in your program too. I heard lead last year and joined your program. So it seems, from here, it seems like you're doing science and you're using the data that you're collecting to advance knowledge. As a consumer in your program, it seems like, you know, they're trying to get you to run more or whatever. And are you going to start to share more of that with the people who are in the program?
Definitely. So we have both a scientific team and a clinical team. And for example, the TMAO score was a discovery that our scientific team did by looking at correlations between cardiovascular health and relationships, the correlation between what's going on in the gut microbiome. Our tagline is scientific wellness, and so we're trying to be wickedly thoughtful around making sure that the data actually stands up, and we have whole protocols around that. But part of what we're doing now is a number of clinical trials. So we have a clinical trial related in Alzheimer's, a second in breast cancer survivors, a third in oral health, where we're working with health partners to actually try to take these dense dynamic data clouds and work with certain disease states. So I appreciate our responsibility is to move into disease states that actually can be very powerful, and our objective is to do that next year.
Yeah, and we may not have time to get into this, but the whole role of machine learning and more advanced analytics, you know, has become a really important part of this. And part of what you get with Arivale is you get these little circles, which we're all going to start seeing, and they're correlations around the edge, or metabolites, the microbiome, all these different tests and chemicals. And you know, there's swirly lines in there that are, you know, it's kind of fun as a consumer or a patient. It makes these correlations. And that's in its infancy, really, to understand all of that, because you can get more and more complicated. But you know, as Ben said, these multi-layers, and you know, should probably even mention that, I mean, there's scanning technologies. I did HLI down in San Diego, and you know, you've got MRI scans, which the average person isn't going to do because they're very expensive. But there are a lot of technologies here that can weigh in on this. So, question?
Hi, I think Clayton knows that both my wife and I have benefited greatly over the last 3 years. Last night we got a tip from Dennis Bushnell that NAD+ was beneficial for us. You're doing a lot of research that doesn't reach the normal Arivale member yet. What I'd like to ask is whether or not you're at a conference here where we would like to hear the latest and greatest things that are about to pop. Is there something that you'd like to share with us? I'll tell you that when I asked about NAD+ for my coach, the answer was at best a neutral on that, because of course you don't have anything that positively supports it, or I think you don't have anything negatively. So maybe you can comment on that and other things.
So our business model that we've embraced is very much the practice of medicine. And as you know, if you're a cutting edge company, the first thing the FDA does is try to shut you down. And so what we've done is embrace the practice of medicine in that a physician is actually ordering all the clinical lab data, and the data goes back to the physician group for a quick review. Our coaches right now are licensed professional dietitians, so they're coaching within their scope of practice. But in 2019, we're going to be bringing on a number of other allied health professionals that'll have the license to talk about a number of other items. In terms of what I think is most exciting in some of the work we're doing is that we're creating polygenic profiles for wellness conditions and disease states. So what does that mean? So I'll give you a wellness example. Right now, you may not know, but if you're in the Arivale program, we've curated 900 genetic variants related to your likelihood of having high LDL, the bad cholesterol. And then what's fascinating is we took people's genetic predisposition, put them into 5 quintiles, and their LDL mapped beautifully to their genetic predisposition unless they're on a statin. So then the coach can be much more sophisticated, and why it seems simple, in that example, if you come in and have high LDL and you have brilliant genetic predisposition, we know behavior lifestyle is going to be highly, highly impactful. Where if on the flip side, you're in quintile 5 and you're disadvantaged, the coach might be saying, have a conversation with your physician about a calcium heart scan. Now we're moving into disease states, and because we've got to embrace the regulatory environment, we can't share the data, but we've done one for Alzheimer's. Correlated 1,400 genetic variants related to someone's likelihood of having Alzheimer's. So of course, what's interesting is we hear about a single genetic variant normally in that disease state, which is rare. And Lee's hypothesis is Alzheimer's is probably 5 to 6, 7 diseases. So we've partnered with the Hoag Hospital. We've taken 200 individuals that are early in the transition into the disease state, creating these dense dynamic data clouds for these individuals, understand their polygenic risk predisposition, and then correlating that back to all the other omics to say, okay, what is the omic that's actually an indication the disease is triggered, and how would we target that? So those are some of, for our perspective, big ideas we hope to be rolling out in 2019. And thank you very much for being in the program.
So Ben, help us out with, in the last couple of minutes, where is this all headed? You're in the research side of it. You're dealing with more the mechanisms of how all this works. Maybe translate what he just said into where we're headed mechanistically and what we're learning.
So I think gene editing is sort of the tip of the iceberg. It's something that's going to work really well to eliminate monogenic disease, but for much more complex disease states, which is the vast majority of them, like Alzheimer's, like hypertension, like arteriosclerosis, it's not a monolocous thing, right? We can't go in and change a gene in your lung and fundamentally alter your health. And so it's going to be multifactorial interventions, right? How can you edit the genome of a particular microbial clade in the human gut in situ to get it to quit producing TMAO when you eat red meat, right? Interventions like that, I think, are the future where we're headed. And also regulatory interventions, not in the sense of gene regulation as opposed to medical regulation, although that would be really nice too. But it's not just editing a gene, changing genetic code. It's really achieving rheostatic control. And in fruit flies and mice and human cell lines, with CRISPR now, we do have systems for achieving rheostatic control. So you don't just have to delete a gene or replace a gene or edit the genome in any way. You can dock a molecule that will simply turn up the volume on a gene in a particular tissue context. And so if you need to fight asthma by increasing epinephrine and smooth muscle tissue in the lung, you can do that, right? And that's where it's going, toward more complex, more sophisticated control over systems. So the future of gene editing is to move beyond gene editing.
No, it's pretty fascinating. And just, I'll mention something real quickly, but I've been doing a project on epigenetics recently, and that's where different chemistry in the body outside of the genome actually activates or turns off cells. And these can be passed down to other generations. And there's some fascinating work being done on around, say, depression and social anxiety that somebody whose grandfather or great-grandfather, there's been some studies out of the Holocaust, and it's these changes that gene expression, and it shows up in later generations. This is early stuff, but there's also talk about being able to actually impact that. So these are dramatic new potential treatments way off in the future in that case, but for things like even depression, which have been difficult to figure out genetically otherwise. So anyway, thank you guys, really appreciate it.
I have an invitation for all of you. I'd love to have you in the program, and given how sophisticated this audience is, offering you 50% off. So if you go to Arivale, normally it's $199 a month. You can stay in as many months as you like. Month 1, you get a coach, you get the genetic data and the blood biomarkers, and you could do that for $99. So when you check out, put Lewis in as your coupon code, and I encourage you all to try the program and let me know what you think. Thank you.
I'm doing it.
Excellent.